Welcome to the Pharma Voice Editor's Take video series, filmed live at the 2012 DIA Annual Meeting in Philadelphia. Hosted by Taryn Grome, editor of Pharma Voice. In this session, Taryn meets with Dr. Cyril Clarke, Medical and Scientific Vice President, Clinical Pharmacology at ICON.
Good morning, Cyril. Welcome to the annual DIA conference and our Editor's Take video program.
Thanks very much. It's good to be here in Philly. It's the first time I've actually attended DIA.
Well, welcome. What do you think are the implications for drug development in the evolving personalized medicine strategy?
I think it's quite a big question to answer in a short period of time, but I think the basis of personalised medicine is really to give the right drug at the right dose to the right person. But I think underlying that now, everyone's looking to make sure that their drug's reimbursed. So I think that pricing strategy really needs to be incorporated from early on. In terms of how you actually develop a drug, I think what we're looking to understand is really the relationship between exposure, response, and the differentiation between desired response and undesired responses. And I think that's leading to people really thinking about what's happening with populations in their trials. And there's really two main pushes at the moment. Historically, we've looked at populations that we've already given drugs to and then tried to look for sub-cohorts that have got better response or better safety, or conversely, adverse response or impaired safety. I think what we're really trying to do now, and especially outside the oncology arena, because the oncology folks have got their act together earlier than I think other therapeutic areas, is to fill trials with people that you predict will respond well to your drug because of inherent characteristics, be they genetic, phenotypic or disease stratifying. And I think that's really leading to a change in how CROs are structured, and I think sort of taking ICON as an example, you see that we're putting a lot of emphasis on really early phase development through our IDS division. We're really trying to put a lot of front-loaded scientific thinking. But then as we pull through into the clinical execution phases, the desire to go quicker and with higher quality data means that we're having to deploy things like our Firecrest and our Iconic technology to make sure that sites really produce very high quality data. And then our late phase group, rather than just concentrating, if you like, on late phase outcomes, is really looking at patient outcomes and what we can assess at the POC stage. So I think there's a lot in the mix in personalised medicine, and we're trying to cover all of that, but it's a space that's not static. And I think it's basically, you need to be science driven in drug development, and that's very much what we're trying to do.
Fantastic. Now let's talk a little bit about what are the key safety related differences between biologics and small molecules.
Well, it's a very, very topical question and quite a tough one to answer in a short space of time. I think we're generally seeing information emerge that registration rates for biologics are better than they are for small chemical entities, and there's probably a number of reasons for that. We're pretty confident in what the pharmacokinetic characteristics of large molecules given parentally will be. With NCEs, we don't always understand as well as we could do what absorption will be. We don't understand metabolism as well as we could do, though we have made progress in having fewer drugs fail due to poor pharmacokinetics. The metabolic liability of large molecules means that characteristically they don't cause hepatic or renal toxicity, and really the safety is driven by exaggerated desired pharmacology rather than off-target pharmacology, though there obviously have been some notable exceptions.
Now, what are some of the key differences between biosimilars and small molecule drug development? I know, a big question in a small amount of time.
I think with a biosimilar, by definition, you've already got proof of concept and you know what the drug has to do because you're trying to replicate the label statement of the originator. So therefore your package is very much focused on, can you show a sufficient degree of similarity to achieve registration? I think when you're looking at small molecules in the generic sense, it's a much more straightforward exercise. The challenge of biosimilars is probably 75% or more of the registration burden occurs before you get into man, in terms of being able to characterise the manufacture, the control and the tertiary structure of the molecule.
Thank you so much for providing your expert insights on these topics.
Well, thanks very much for inviting me to the Pharma Voice booth, It's something that I've always followed, and I've really enjoyed DIA and I've enjoyed Philadelphia.
Additional Editors Take videos, as well as podcasts, white papers, webinars, and more, are available in the resources area at www.pharmavoice.com.